Histochemical and immunotechniques. Antibody generation. The antibody or Y-shaped protein produced by the plasma cell in response to pathogens and acts as a major effector of immunity. Antibody are also known as immunoglobulin IgG and recognizes unique molecules of any foreign body termed as antigen. These antibody have many utility in medical research. These molecules generated by the immune specifically fit a target within the body and are often used as a biochemical markers or as a method of targeting drugs as well as to study infectious agent or allergies. All vaccines research relies on the use of antibody, and they are frequently used for in vitro or culture work and provide a valuable tool for identifying or locating any specific structure found in the blood. The antibody cannot be made artificial means and must be generated by an intact mammalian immune system. Many animals can be used to generate antibody, including sheep, goats, dog and horses. Antibody generated in rabbits. While rabbits are often used for generation of antibody in laboratory conditions, the rabbit used in antibody production are generally fully grown animals of over 12 weeks old and are kept in a facility for no more than 6 months. 1. A test bleed is usually carried out on the rabbit before antibody production is initiated. For this process, animals are given a sedative such as azithromycin. The syringe and needle are then inserted into the marginal vein so that the needle is visible inside the vein. When the plugger pulls back slowly to take blood from the ear, the blood can be seen filling the syringe. 2. When the desired amount of blood has been collected, the syringe is carefully removed and pressure is applied to the vein until the bleeding stops. The blood is then stored in the serum tube, which is kept at 37 degrees centigrade until the blood clots and then refrigerated. 3. Once the test bleed has been performed, the antibody production can begin by immunizing the rabbit. The area to be injected is shaved and cleaned with alcohol. The antigen to which the antibody will be produced is injected subcutaneously beneath the skin, usually in several different areas along with adjuvant. A substance designed to increase the immune responses so that more antibody are produced. 4. The rabbit is then left to recover and produce antibody. The test bleeds are carried out at regular intervals to check the level of antibody in the blood. Once the appropriate level of antibody has been reached, the blood is removed from the rabbit using a cardiac bleed. 5. In this procedure, rabbit is deeply anesthetized and the needle is inserted directly into the heart and it is beating so to remove as much blood as possible from the body. Generally, more than 100 ml of blood can be obtained from each rabbit. When no more blood can be obtained, the rabbit is killed by a lethal injection of pentobarbital into the heart. 6. The aim of this procedure is to generate high titer, high affinity anti-serum that can be used in experiments or diagnosis of illness or allergy. The whole procedure may take several months, but the time will vary depending on the antigen and the animal used. Adjuvants. The adjuvants are the substances that enhance the immunogenicity of an antigen. When they are mixed together and injected, they are used mainly to boost the immune response of antigen having low immunogenicity or is present in very limited amount. The effect of adjuvants are 1. Prolonging of antigen persistence. 2. Enhancement of co-stimulatory signals. 3. Increase of local inflammation. 4. Stimulation of non-specific proliferation of lymphocytes. One examples of such adjuvant is aluminum potassium sulfate, which when mixed with an antigen, it causes the antigen to precipitate and results in slow release from the site of injection. This release of effective time of exposure of antigen from few days to several weeks and also increases the size that aids in their phagocytosis. The Frown's complete adjuvant is the first deliberating science highly effective adjuvants developed by the Julius Frown. This is a water in oil adjuvant and contains heat-killed mycobacteria as an additional ingredient. The muramyl dipeptide, a compound of mycobacterial cell wall, activates macrophages, giving its characteristics strong immunogenicity. A variation known as a frayed incomplete adjuvant contains antigen in aqueous solution, mineral oil, and an emulsifying agent such as a mannate monooleate, which disperses the oil into the small droplets surrounding the antigen. The antigen is then released very slowly from the site of injection. Radioimmunoassay RIA. It was developed in 1960 by S.A. Berson and R. Rallo. The radioimmunoassay RIA is one of the most sensitive techniques for detection of antibody and antigen. The radioimmunoassay utilizes a competitive binding of radio-labeled antigen and unlabeled antigen to a high affinity antibody. The labeled antigen is mixed with the antibody at a concentration that saturates the antigen binding sites of the antibody. Then test samples of unlabeled antigen of unknown concentration are added in progressively large amount. The antibody does not distinguish labeled from unlabeled antigen, so the two kinds of antigen compete for available binding sites on the antibody. As the concentration of unlabeled antigen increases, more labeled antigen will be displaced from the binding site. The decrease in the amount of radio-labeled antigen bound to specific antibody in presence of the test sample is measured in order to determine the amount of antigen present in the test sample. 1. Radio labeling of antigen is usually performed by using gamma emitting isotopes like iodine 125 or beta emitting isotopes like hydrogen 3. 2. The next step is to determine the amount of antibody needed to bind 50 to 70% of fixed quantities of radioactive antigen in the assay mixture. This ratio of antibody to antigen is chosen to ensure that the number of epitope presented by the labeled antigen always exceeds the total number of antibody binding sites. 3. The unlabeled test antigen added to the sample mixture then competes with the radio labeled antigen for the limited binding site available on antibody. Even a small amount of unlabeled antigen added to the assay mixture of a labeled antigen and antibody will cause a decrease in the amount of radioactive antigen bound, and this decreases will be proportional to the amount of unlabeled antigen added. 4. Determination of amount of labeled antigen bound to form the antigen antibody complex is precipitated to separate it from free antigen, and the radioactivity is precipitate is measured. A standard curve can be generated using unlabeled antigen sample of known concentration, and from this plot the amount of antigen in the test mixture may be precisely determined. Nowadays the separation of antigen antibody complexes have been made easier by covalently cross-linked the antibody with separose beads, thus creating a solid phase radioimmunoassay. The amount of radio-labeled antigen bound to the beads can be measured after the beads have been centrifuged and washed. Alternatively, the antibody can be immobilized on polystyrene or polyvinyl chloride wells, and the amount of free labeled antigen in the supernatant can be determined in a radiation counter. A more popular approach is to immobilize the antibody to microtiter wells. This procedure works well when the considerable greater number of antigen to be assayed, as a typical microtiter plate has 96 wells. ELISA enzyme linked immunosorbent assay. The ELISA approaches is detected for direction of a specific antigen is very similar to radioimmunoassay, but utilizes enzyme based catalysis to facilitate detection. The basic principle of this process is to allow an enzyme conjugate antibody to react with a colorless substrate in order to produce a color product. Such substrate are termed as a chromogenic substrate. Some commonly used enzymes for ELISA or alkaline phosphatase, horseradish peroxidase, and alpha galactosidase. The variety of ELISA methodologies have been developed to facilitate a wide range of directions. Indirect ELISA. The quantitative determination of antibody is performed by indirect ELISA. This method is widely used to detect the presence of serum antibody against human immunodeficiency virus HIV and can be done within 6 weeks of infection. 1. The serum or some other sample containing the primary antibody AB1 is added to an antigen-coated microtiter well and allowed to react with the antigen attached to the well. 2. After any free antibody 1 is washed away, the presence of antibody bound to the antigen is detected by adding an enzyme conjugated secondary anti-isotype antibody AB2 which binds to the primary antibody. 3. Any free antibody 2 then is washed away and the substrate for the enzyme is added. The amount of the color reaction product that formed is measured by specialized spectrophotometric plate reader, which can be measured the absorbance of all the wells of 96 plates in seconds. Sandwich ELISA. 1. In this method, antibody is attached to the microtiter plate and a sample containing the antigen in question is added to allow reaction with the bound antibody. 2. The well is then washed and then a second enzyme linked antibody specific for different epitopes of antigen is added. 3. Any unbound secondary antibody is removed by washing and then a chromogenic substrate is added to develop color in presence of antigen. Competitive ELISA. For competitive ELISA, the antibody is incubated in the solution with a sample containing antigen. 2. The solution now containing the antigen antibody complex is now added to the microtiter well, which is already coated with the antigen. 3. Any free antibody present within the solution will bind to the antigen coated well. 4. The addition of of an enzyme conjugated secondary antibody AB2 specific for the isotope of primary antibody can be used to determine the amount of primary antibody bound to the well. Western blotting. The Western blotting, as described earlier, provides a method for separating specific protein from a mixture of different proteins. 1. In Western blotting, the protein mixture is electrophoretically separated on an SDS polyacrylamide gel SDS phase. 2. The protein band are transferred to a nylon membrane by electrophoresis and the individual protein bands are identified by blending the nitrocellular membrane with the radio-labeled or enzyme-linked polyclonal or monoclonal antibody specific for the protein of interest. 3. The antigen antibody complexes that forms on the band containing the protein recognized by the antibody can be visualized in a variety of ways. If the protein of interest was bound by a radioactive antibody, its position on the blot can be determined by the autoradiography. However, the most generally used detection procedure employed enzyme-linked antibody against the protein. Immunoprecipitation. The isolation of antigen for further investigation is carried out using an immunoprecipitation assay. In order to perform this assay, 1. The cell or tissue are ruptured and their extract is mixed with an antibody designed to target that antigen. This leads to the formation of antigen antibody complex leading to precipitation of antigen of interest. 2. However, if the concentration of antigen present is low, the procedure can take much more time to form any significant immunoprecipitation. 3. This problem can be avoided by either attaching the antibody to solid medium, allowing immunoprecipitation by centrifugation, or by adding secondary antibody designed to bind the primary antibody. 4. The immunoprecipitation has also been used to determine whether the synthesis of antibody happening from a cell or a tissue. This is done by biosynthesis radioisotope labeling. 5. Radioisotope labeling of protein from cell of interest can be done by growing the cell in a cell culture medium that has one or more radio-labeled and amino acids such as leucine, cysteine, and methionine, as there are less susceptible to metabolic modification. Following growth, these cells are lysed and antibody designed against the target antigen are added. 6. The resulting antigen antibody complex is collected. NY immunoprecipitation is washed and then analyzed using a scintillation counter to get a quantitative estimation of the amount of the protein produced by the cells. Immunofluorescence. The procedure of immunofluorescence was first developed by Albert Kunz in 1944, who was able to label antibodies with fluorescent molecules. The fluorescent molecules absorb light at one wavelength by a process termed excitation, and then release light at a different wavelength or by emission. The antibody molecules tagged with the fluorescent dye can be used to visualize antigen antibody complex. The visualization is carried out by using fluorescent microscope, which uses a UV light source. Some common fluorescent dye used for the purpose are one fluorescein. They absorb blue light at 490 nanometer and emits yellow green light at 517 nanometer. To rhodamine absorbs yellow green range 515 nanometer and emits deep red fluorescent 546 nanometer. These two dyes can be used to perform two color immunofluorescence assay. In this method, an antibody specific to one determinant is labeled with fluorescent, and the antibody recognizes a different antigen is labeled with rhodamine. The location of the fluorescent tagged antibody will be visible by its yellow green color, easy to distinguish from the red color emitted where the rhodamine tagged antibody has found. By conjugating fluorescein to one antibody and rhodamine to another antibody, one can for example visualize simultaneous two different cell membrane antigen on the same cell. Fluorescent tagged of antibody can be performed by two ways: direct staining. The antibody is directly conjugated with the fluorescein. To indirect staining, the primary antibody is unlabeled and it is detected with an additional fluorochrome labeled reagent. The indirect immunofluorescence staining has two advantages over direct staining. First, the primary antibody does not need to be conjugated with the fluorochrome because the supply of primary antibody is often a limiting factor. The indirect method avoids the loss of antibody that usually occurs during the conjugation reaction. The second indirect method increases the sensitivity of staining because multiple molecules of fluorochrome reagent binds to each primary antibody molecules, increasing the amount of light emitted at the location of each primary antibody molecules. Flow cytometer. The quantitative data based on the fluorescence method is achieved by developing the flow cytometer that automates the analysis fluorescent tagged cell. 1. The flow cytometer uses a laser beam and light detector to count single intact cell in suspension. During the passage of cell through the laser beam, the light from the detector is deflected and this deflection is recorded. 2. Those cells having a fluorescent tagged antibody bound to their cell surface antigen are excited by the laser and emits light that is recorded by the second detector system located at the right angle to the laser beam. 3. The simplest form of the instrument count each cell as it passes the laser beam and records the level of the fluorescence the cell emits. An attached computer generates the photo of the number of cells in one axis and their fluorescence intensity in the other. More sophisticated versions of instrument are capable of sorting the populations of cells into different containers according to their fluorescence profile. Immunoelectron microscope. The fine specificity of antibody has been used for visualizing specific intracellular tissue components by immunoelectron microscopy. In this process, an electron-dense label is either conjugated to a specific antibody or direct staining or conjugated to an anti-immunoglobulin reagent for indirect staining. 2. The number of electron dense labels have been used such as colloidal gold because the electron dense label absorbs electron. It can be visualized with the electron microscope as small black dots. 2. In case of immunogold labeling, different antibody can be conjugated with the gold particles of different sizes, allowing identification of different antigen within a cell by different size of electron dense gold particles attached to the antibody. Detection of protein within a cell. One popular approach of detecting molecules within a cell is the use of fluorescent property of photon. When a photon is observed, an electron in the observing molecule chromophore is lifted to a higher energy level. This is an all or nothing event. To be observed, the photon must contain a quantity of energy, a quantum that exactly matches the energy of electronic transition. A molecules that have observed a photon is in an excited state, which is generally unstable. An electron lifted into a higher energy orbital usually returns rapidly to its normal lower energy orbital. The excited molecules delay to be stable ground state, giving up the absorbed quantum as light or heat, or using it to do chemical work. The light emission accompanying decay of excited molecules called fluorescent is always at a longer wavelength, lower energy than that of absorbed light. The fluorescent probe are regularly used to detect changes happen within the cell. The probes are usually used to generate real-time information regarding the concentration of protein and activities of enzymes like protein kinase. The localization of protein function can also be performed using this approach. One of the most commonly used fluorescent tag is derived from the green fluorescent protein GFP of the jellyfish Aquario Victoria. When excited by absorption of a photon of light, the green fluorescent protein emits light in the green region of the spectrum. 2. The green fluorescent protein has a 11 standard barrel structure, and the light absorbing emitting center of the protein comprises the tripeptide serum 65, tyrosine 66, and glycine 67. 3. The variant of this protein with different fluorescent spectra can be produced by genetic engineering of green fluorescent protein gene. A yellow fluorescent protein YFP is produced by replacing the alanine 206 in green fluorescent protein by a lysine residue, changing the wavelength of light absorption and fluorescence. Other variant of a green fluorescent protein fluorescence blue BFP and cyanine CFP light and a leaded protein MRFP1 fluorescence red light. Fluorescence resonance energy transfer, or FRET. The fluorescence resonance energy transfer FRET is a method to identify the protein interaction within cell. 1. In this method, the energy of excited molecules, the donor, passes directly to a nearby molecules, the acceptor, without emission of photon exciting the acceptor. 2. The acceptor then returns to its ground state by emitting the photon having longer wavelength than the original exciting light and the fluorescence emission of the donor. This second mode of decay, fluorescence resonance energy transfer, is possible only when the donor and acceptor are close to each other within 1 to 50 Armstrong. 3. Thus, very small changes in the distance between the donor and acceptor register as very large changes in fluorescent resonance energy transfer, measured as the fluorescence of the acceptor molecules when the donor is excited. With the sufficiently sensitive high light detector, this fluorescence signal can be located to specific region of a single living cells. 4. The fluorescence resonance energy transfer has been used to measure the CAMP concentration in the living cells. The gene for the GFT is fused with that for regulatory subunit of CAMP dependent protein kinase, and the gene for BFP is fused with that for catalytic subunit. When these two hybrid proteins are expressed in the cell, the BFP donor excited at 380 nanometer, emission at 460 nanometer, and GFP acceptor emission at 475 nanometer, emission at 545 nanometer. In the inactive protein kinase A are close enough to undergo fluorescence resonance energy transfer. Fluorescence recovery after photo bleaching FRAP. The rate of fluorescence recovery after photo bleaching FRAP is measured the rate of lateral diffusion of fluorescence tagging lipids. Using the fluorescence recovery after photo bleaching techniques, movement of lipids and proteins on the cell membrane can be observed and the rate of their movement measured. 1. The lipid in the lower leaflet of the plasma membrane are labeled by reaction with the membrane impairment fluorescent probe red, so the surface is uniformly labeled when viewed with fluorescent microscope. 2. A small area is bleached by irradiation with an intense laser beam, leaving that area non-fluorescent. 3. With the passage of time, labeled lipid molecules diffuses into the bleached region and it again becomes fluorescent. 4. From the time course of fluorescence return to its area, the diffusion coefficient for the labeled lipid is determined. 5. The rates are found to be typically high. The lipid movement at this speed could circumnavigate an E. coli cell in 1 second. Fluorescence in situ hybridization FISH. Before genome sequence become a common method, several thousand human genes have been already mapped to specific location within the human chromosome. One common method used to obtain this data is through in situ hybridization of a fluorescent dye to chromosome. This method is known as a fluorescent in situ hybridization. In situ hybridization usually carried out on a metaphase cell, as this allows the mapping of a cloned gene to a locus defined by chromosome band. However, as each band of human chromosomes contain thousands of kilobases of DNA, this method does not provide detailed mapping information. The polythene chromosome of Drosophila allows localization of gene to interpret the chromosome band containing only 10 to 20 kilobases of DNA. Higher resolution can be obtained by hybridization to more extended chromosomes during the pro-metaphase or interphase cells. This allows FISH mapping of a cloned gene to region of about 100 kilobases. The fluorescence in situ hybridization can also be used to detect and localize a specific RNA target, mRNA, INC RNA and miRNA in cells, circulating tumor cells and tissue samples. In this context, it can be help define the spatial temporal pattern of a gene expression within the cell and tissue. GISH, genomic in situ hybridization. When total genomic DNA, often consisting of entire nuclear DNA of a plant, is used as a probe in hybridization experiment to chromosomal DNA in situ, the method is called as genomic in situ hybridization (GISH). The repeated sequence that makes up 40 to 95% of plant genome is in higher plants reannul more rapidly than the unique sequences. The genomic hybridization method investigates the interspecies distribution and organization of these sequences. The method involves extraction of genomic DNA from one of the species of plant to be used as a probe for in situ hybridization to chromosomes of a plant or hybrid being studied. Many of the DNA sequences within the genomes under investigation may be sufficiently different to be investigated by GISH. Genomic in situ hybridization. The genomic in situ hybridization permits characterization of a genome and chromosome of hybrid plants, allow polyploid plants and recombinant breeding lines.